Right, okay, I'm going to get started so that we stay on time. So welcome everybody. Thank you so much for signing up to our webinar. This is one of 4 webinars, which I know a lot of you have signed up to more than one of them. And today is all about the future of elm and the research that we've been doing at Forest Research and at Kew on British elm. So this is the programme today. We're going to be starting with Joan from Forest Research talking about beetle behaviour, and then we'll be looking at mapping of UK elm, followed by 2 talks on the genomics, and then finishing with a talk about land manager perceptions from a social science project. And then after the break, we'll be having a panel discussion with the questions from you. So a little bit about the mission for the Centre for Forest Protection, in case you're not familiar with it. The Centre is a research collaboration that's funded by DEFRA, and it's led by Forest Research and Kew. And the focus is very much on the future of forests and using innovative science and interdisciplinary and collaborative research. Every year as well, we offer some 6-month internships at Forest Research and Kew working on Centre for Forest Protection projects. So the interns work on a live project and they really contribute to the research, and you'll be hearing from 3 of them today about the projects that they've been doing this year. If you want to find out more about forest protection, we have the Centre for Forest Protection, we have a website, and information about all of our projects is on there. We're just coming to the end of our first phase, and we will be starting a new set of projects in April. So take a look at the website to find out more about those. So the most of the talks today are from a joint project between Forest Research and Kew on the distribution and diversity of existing UK elms. And we're going to start with a talk by Joan Webber, who is a researcher at Forest Research, one of our senior researchers who's been working on Dutch elm disease for years and is a real expert on the pathology and on the beetle. So I'm going to hand over to Joan, who's going to be talking about her research over the last couple of years on beetle behaviour and the performance of Dutch elm resistant elms. Over to you, Joan.
Well, thank you, and I hope good morning to everybody, and I hope you can see that first slide. And yes, this was a small focus of a larger project looking at diversity of elms, and we really wanted to look at beetle behaviour and the performance of some of these disease-resistant elms that have already been planted out in the countryside. And this is some work that I undertook with my colleague Clive Brazier, and I also really want to acknowledge the support and help that we've also had from Andrew Brooks at the East Hampshire Butterfly Conservation organisation, because it's really thanks to some of the work that Andrew Brooks has done planting out these elms that we've been able to look at them and see how they are performing in the field. So we had 2 really main areas of focus, and one that I briefly touched on then. These trees that have already been planted out, these often resistant elm cultivars, either come from breeding programmes or considered to be resistant from the way they perform in the field. And we wanted to look at them under Dutch elm disease pressure and see what their outcomes were. And the other element of the project that I'm going to deal with a little bit later is looking at the presence and the relative frequency of the 2 bark beetles that act as vectors of the Dutch elm disease pathogen Ophiostoma novoalmi. And I thought, just as a little reminder to everybody, I would just touch on the Dutch elm disease cycle as well. And for the purposes of what I'm talking about today, it's really worthwhile considering the disease cycle in terms of what happens with the healthy elms and then what happens when they're diseased. And really, when it comes to beetle behaviour, the important thing is what goes on with these healthy elms, because when beetles are dispersing, which will be a little later in the year, in April, May, spring, early summer, they emerge from their breeding materials and they disperse and fly. And as they're flying, they have to make pit stops to stop themselves becoming too desiccated. And to do that, they feed in the crowns of healthy elms. And they just feed in the upper part of the crown, in the crotches that are made between twigs that are maybe a couple of years old, 2 or 3 years old, as they are subtended from slightly older twigs, and they chew away the bark. And that's what you can see in that uppermost picture there, that beetle in that feeding twig crotch, and where it's chewed away that bark and exposed the xylem, as you can see there. And that is the moment where, if the Dutch elm disease pathogen, Ophiostoma novo-ulmi, is carried by that beetle, it can be transferred into that wound, and that's how it gets into the vascular tissue of the tree. And then you get tylosis response from the tree with this streaking or blackened dots that you can see there. And that is as the fungus moves around in the tree, and the tree is responding to the fungus. Those beetles don't sort of hang around. This process of feeding is a matter of hours. Sometimes beetles will remain in those feeding grooves for maybe a day or 2 days if the weather isn't very conducive to flight and dispersal. But they will then fly off, and really it's an unknown quantity what happens, because only a small number of those feeding grooves will actually result in the fungus, the pathogen, gaining access to that xylem. But once it's in that tree, then really the prognosis tends not to be very good for that tree. And then other beetles, while they're dispersing as well, will also be looking for these declining trees to breed in, and they will then invade the bark. And when they go into the bark, if they're still carrying the fungus, they introduce the fungus into that bark, where the beetles will breed. Larvae will develop into a new brood of beetles, which eventually emerge the following year. And while they're in that bark, they will pick up the fungus as well. So this process of feeding, which isn't obligatory for these bark beetles, is really an unfortunate side event which can allow the trees to become diseased. So when we're thinking about, well, how are these resistant elms performing under Dutch elm disease pressure? The way we approached it was to try and quantify the amount of feeding by these beetles on individual trees in the field. And what you can see in that right-hand picture down the side of that slide, I don't know if I move my cursor, you can see here is where a beetle has chewed away that bark and made a feeding groove or a feeding wound, and that has started to callus over. But you can get all these potential feeding positions, count where they are, and look to see the number where there are signs of feeding going on. So we did this with trees in the field, and our other way of looking at the performance of resistant elms was to try and find out where there were any records that people had of when trees had become resistant trees had become infected by Dutch elm disease, and try and get an idea of the number of trees and how extensive the symptoms were. So, working particularly with Andrew Brooks here, we focus pretty much on one side. And as you can see, there's quite a range of different elms that come from different research, different breeding programs really around the world, but mainly Europe and some from North America. But our initial problems when trying to do this work were having enough of different sorts of elm trees so we could make comparisons. So at the end of the day, we focused on feeding behaviour of the beetles on one clone of Ulmus minor, Adamus, which has relatively recently been released from a Spanish breeding programme. And these Ulmus minor are similar to the sort of clones of elms that we would see in the countryside here in Britain. And we also looked at the beetle feeding on that, and also Ulmus laevis, which is European white elm, which has been promoted very much over the past few years of an elm to plant and an elm to think about because of the way it seems to perform in the field. And very often you will find people will tell you that it is resistant. And so what we tried to do was find trees that were growing in close proximity, an adamus and a laevis. So the assumption could be that they were really exposed to similar numbers of beetles dispersing and potentially feeding on them, and then look to see if there were any similarities or differences in that feeding. And we tried to do this at different distances away from beetle sources, because as beetles emerge and fly, they fly towards the light, and their numbers will gradually be diluted over larger and larger distances. So this just gives you a little glimpse of some of the results that we were getting. And the blue bars are the Ulmus laevis, and the orange bars are the Ulmus adamus. So the Ulmus laevis versus the Ulmus minor. And what we're seeing usually with the bars on the left-hand side of the graph is where they're quite distant from a beetle source, and then usually with the taller bars where they're closer to a beetle source. And so one of the first things that we noticed was that, generally speaking, where we were comparing the Ulmus laevis with the Ulmus minor, the adamus, the beetles preferred to feed on the adamus, the Ulmus minor, much more than the Ulmus laevis. And that's consistent with what other people have shown and noticed in the field. And that seems to be one of the reasons why Ulmus laevis seems to be able to perform well in the field where there is some Dutch elm disease around. But what we found was, once the trees were exposed to a source of beetles that were very close by, then really that difference in their preference to feed on the field elm over the white elm became really quite not really noticeable. And you can see that, for example, with graph C here, and we're looking at three types of trees here, a laevis here, and then an adamus, and then this is vada, another resistant elm, and the amount of feeding on the three of them is about the same, and they're very close to a beetle source. Whereas if you're looking at beetles that are more coming from a more distant source, and you're comparing between, say, adamus and laevis, which is on the left-hand side, you tend to get this differential. So usually laevis is less preferred for feeding, but that preference to go for a field elm over a laevis tends to become blurred once the beetles are very close by. So what we found when we are looking at these paired trees, what I've already said, that the extent of beetle breeding depends on the distance from a beetle source. Also the weather, we have periods where the weather was very conducive to beetle flight, it synchronized it, and you would get a lot of beetle feeding going on, and of both beetle species, Scolytus scolytus and Scolytus multistriatus. Usually the laevis was less preferred to feed on, but even elms that tend to be less attracted to the beetles for feeding can end up with heavy feeding when those beetle populations spike up. But one of the things that was most interesting was, whereas we saw with the resistant elms, adamus and vada, that have been released from elm breeding programs and challenged over that process of breeding with the Dutch elm disease pathogen, they never developed any external symptoms despite very heavy feeding, particularly in 2022 and 2023. But we did see, intriguingly, with adamus, this Ulmus minor clone that is resistant, never developed any external symptoms, but that didn't mean it didn't become infected. We could see signs of tylosis and streaking in the xylem vessels. So it did become infected, but it seemed to be tolerant of that infection and didn't develop symptoms. This really was in contrast with laevis, which, when the amount of feeding on Ulmus laevis began to increase, we did see dying Ulmus laevis lavis trees, and then they became used for breeding by the beetles, and that added to the number of beetle sources as well. So just turning to this counterpoint of looking at the performance of resistant elm cultivars as reported to us, or where we could find records, we did find some records of some elms considered to be resistant that had often come from breeding programs. And in some cases, they would develop some defoliation, and then they would usually, most often, recover the following year. Although we did have a couple of trees, for example, Retiro and Christina Buisman, which is quite an old release from a breeding program, that did go on to develop a lot of defoliation and either had to be died later on or had to be removed. And also with this resistant version of Ulmus americana, Princetown as well. So we do see some failures in some of these resistant elm cultivars that have been released from some of these breeding programs, but by and large, even if they become infected, they usually recover, and the infection is quite minor. So they're not immune. Quite a lot of them can become infected, probably when there are large numbers of beetles around feeding, but for the most part, they recover. So I want to turn a little more about beetle feeding and Ulmus laevis. And this is really all about the performance of Ulmus laevis and these Ulmus laevis trees that are being planted on some of these butterfly conservation sites. And we were lucky enough to be able to look at about 50 of these Ulmus laevis planted out because of their field performance, some of them about 20 years old now, across a number of different sites. Here's one that has, as you can see, died of the disease, and in fact, the bark has been used for beetle breeding. And these trees had performed very well year after year until we came to about 2022 and 2023, when we've had good summers, a lot of beetle emergence going on, and then we've seen the failure of this particular species of elm tree, Ulmus laevis, succumbing to Dutch elm disease. So performance in the field, the fact that the beetles usually don't like to feed on it, is not sustaining it over the longer term when beetle numbers really start to increase in some areas. And as you can see, out of this total of 50 Ulmus laevis, by the time we got to 2024, something like approaching 70% of them had become diseased or had died of disease. So I think that really put it in perspective that if you want to be sure of the performance of your elm, you have to have it from really a consistent and reputable breeding program, and not really rely on trees that have performed well in the field and assume that they are resistant, because that isn't necessarily the case. Now, I want to touch on fairly briefly the other part of this project, and that was establish the presence and the relative frequency of the two vector beetles, Scolytus scolytus and Scolytus multistriatus. And we did this quite simply by going and visiting a series of sites across southern England, from Devon right through to Sussex, and quantifying the extent of beetle breeding that we could see in those trees as they succumbed to the disease, what species it was, and what impact they were having. And I'm going to show you some of the information that came from 2 sites where we were able to take quite a close look at quite a good number of trees at each site, between 40 and 80 trees at each site, and look in some detail at the beetle breeding. And I suppose one of the things that was sort of maybe mildly surprising was we very much see these peaks and troughs in disease striking, for example, hedgerow trees, and usually it's thought that the trees get to 15 or 20 years old before they succumb. And that usually is the case. And we do see, for example, that they usually, you don't get a local epidemic building up until the trees are big enough and have thick enough bark to sustain breeding beetles. But when we looked at the size class of the elms, and this is just the elms from the 2 different sites, one in Devon and one in Hampshire, what we found was, although we got quite a large number of trees, for example, that had a stem diameter of 20 centimetres or less, we also got some that were well over 40 centimetres in stem diameter, even over 50 centimetres in stem diameter. So some of these trees are surviving quite considerable amounts of time, well beyond the 15 or 20 years that usually considered the norm. And so when they do succumb, they can sustain quite a lot of beetle breeding as well. But I'm just going to really touch on this in more generalities. But where we measured those trees, usually we found both the bark beetles. I think at all sites we found both bark beetles, Scolytus scolytus and Scolytus multistriatus. And that's an interesting observation, and that Scolytus scolytus is by far the better, the more effective vector compared with Scolytus multistriatus. So the suggestion was that as trees haven't been reaching this larger size, that the epidemic that we have now of Dutch elm disease is really dependent on multistriatus. And that really isn't the case. We're finding both of them, but usually the trees have to be at least 20 centimetres diameter before we're seeing Scolytus scolytus really starting to breed in any numbers. And the other thing is, as you watch the epidemic develop over a number of years, these small local epidemics on hedgerow trees or on the edge of woodlands, initially you will get trees being killed. Often they're some of the largest trees being killed, but they're often not found later on by beetles to breed in. So some of these local epidemics get off to quite a stuttering start. So very often it is only with Scolytus multistriatus, and then gradually you get more and more breeding, the trees being used more efficiently for breeding as you get beetles that have been able to establish in those trees on that site, then just flying short distances to infect and breed in others. And in those early stages, though, many trees die with little or no breeding. So they're being found by, if you like, immigrant beetles that are coming from outbreaks further away. It's very much a hit or miss effect. Some become infected, most don't. And then there's very much a hit or miss effect of whether they're then found for beetle breeding and how quickly beetle numbers then tend to build up in those areas. So still ongoing. Well, we're analysing some of these data just to get a better handle on these Scolytus multistriatus ratios, but we're currently writing up a research paper, and we're particularly going to focus on Ulmus laevis as a cautionary tale, which also has relevance to wych elm as well, which performs well in the field, but is very susceptible to the pathogen, but the beetles tend to avoid it when feeding, but that doesn't mean it has a long-term sustainable, or individual trees don't have a long-term sustainable future. Even if the beetles tend to avoid feeding on them, if that is the only tree they can find to feed on, or beetle numbers build up, then I think the same thing that we saw with laevis will happen with Ulmus glabra. And we're going to try and focus a bit more on performance of the disease-resistant elms and see if we can find some more records on where they failed or where they haven't failed. And one thing that we hope to do is look to see how we may have an increasing number of days that are suitable for beetle flight as our climate is warming up, particularly with warmer springs and summer, particularly in Scotland, how that may also be driving the disease in some parts of Britain. And I think that takes me on to my last slide. And I just want to say, really, a lot of the work that we've done in the field looking at these resistant elms, we couldn't have done without the help of Andrew Brooks as well. So thank you.
Thank you so much, Joan. That was really fascinating. Such a lot of work on both those areas there. So our next speaker is Catherine Walter, and Catherine is one of the Centre for Forest Protection interns. who has been working on mapping UK elm and has been working for the last 6 months at Kew contributing to this project. So Catherine, I'm going to hand over to you.
Morning, everyone. As Louise mentioned, I'm Catherine and I'm an intern with the Centre for Forest Protection based at Kew. And for the past 6 months, I've been doing an internship to map the distribution of elm in the UK. So to start with, I'm going to give a quick overview of all the different elms that can be found in the UK. So we have the most commonly found elms, which are the native wych elm, field elm and English elm. And then these two species, Ulmus glabra and Ulmus minor, naturally form hybrids in areas where their range overlaps. So we also have hybrids between these species. And then elm has a long history of being cultivated, leading to the development of many cultivars. And then due to their use as ornamental trees, lots of non-native species and their cultivars have also been planted in the country. And then, of course, with Dutch elm disease in the 20th century, most of the mature elms that were distinctive of the landscape were killed, which has led to elms still being able to persist, but as small recruitment elms that often fail to reach maturity. And so in response, efforts have been made to identify native elms that may be exhibiting resistance, but also to breed resistant elms. So in the UK, we have the British elm species, we have their hybrids, we have elm cultivars, we have introduced species, and now we have disease-resistant cultivars. Here is a timeline that I made of various dates in which these disease-resistant cultivars were either introduced to the UK through trials or elm planting projects, or dates of their commercial release where the introduction date wasn't available. And you can see that over time, a large number of these cultivars have been produced, and those that are highlighted in pink contain Asiatic elms in their ancestry in order to increase the resistance. And I believe Cas is going to be talking in more detail about one of these in particular in her genomics work. So with all of these different types of elms in mind, I set out to gather data for all of the different types of elm in the UK, map their distributions, and then collate this information into a database. I also set out to update records of Sapura autumn gold cultivars that were planted in the 1980s as part of a campaign that distributed them to schools around the country. This would be a great opportunity to collect data on mature cultivars, as many of them are particularly new. This is like a knowledge gap that would help determine their long-term suitability. To do this, I collected data from a variety of sources, including open data repositories, and then also directly communicating with individuals. I then organised the data and filtered it. The taxonomy of elms is very complex, and so you could find many different names being used in different sources. And so I assigned synonyms where I could to make sure that maps represented the same taxa where possible. And then once I'd done this, I removed duplicates based on their name and their location. And then I used this to map the distribution. So in total, over 125,000 records were obtained, and this was reduced to about 80,000 after I'd filtered them. 336 different elm names were used, but then after I applied synonyms, this was 269 distinct elms. You can see from the graph on the right that the majority of these were cultivars, but there was also a large number of hybrids and a large number of species, considering the number of non-native species that have been planted. Although the I think the first record was from 1794. Over half of the records were from the year 2000 onwards, which indicated that the data was representative more of the current state of elms rather than a historical relic. And then some of these elms didn't have any location information. So in the end, I was able to produce maps for 252 different types of elm. I don't have time to go through all 252, so I'm just going to give a summary of the results. So to start with, we have the British elms, and the data agreed with previously described distributions of these different elms, with field elm and English elm more prevalent in the south and southeast England, and then wych elm more frequently recorded as you move northwards. So this was a really great starting point, and the distribution was similar even when you filtered the records for those more recent ones. And then, as I mentioned, there are lots of elm cultivars and non-native species that have been planted in the UK, and the distribution of these showed this nicely, as their distributions were rather sporadic and they were more rare in occurrence, which indicated them being planted in gardens or as street trees. So I've got two examples here, one of a wych elm cultivar, which, although it actually had a fair few number of records for a cultivar, they are kind of all spread out, which shows that they've been planted. And then a North American elm, Ulmus rubra, which has also been planted in parks and gardens in the UK. To follow on from what Joan was saying earlier about Ulmus laevis, although this is an introduced European elm, it has been a more of a wide distribution compared to the other introduced species, and this is because of its incorporation into elm planting projects. So over half of these trees shown on this map here were planted in the last 10 years due to its unattractiveness to the beetle. In addition to Ulmus laevis, lots of disease-resistant cultivars have been trialled and planted in the UK. So the data showed that a total of 73 different types have been introduced. However, over 2/3 of these were of just 5 cultivars, with the highest number of records for Lutece, followed by New Horizon, Adamus, Lobel, and Sapura Autumn Gold. And these seem to have tended to be planted in the south of England, with the highest number of records in Hampshire, followed by Greater London and Brighton and Hove. So I'm going to finish with 2 examples of why collecting this data and mapping these distributions is important. And so to start with, I think that my attempt to update records of Sapura Autumn Gold trees from this campaign in the 1980s demonstrates this nicely. So from the Conservation Foundation's archives, there was a list of 250 schools that were sent these cultivars. I was able to contact 137 of them, largely due to a number closing down or moving site. And then I only had responses from 28, and most of these didn't know anything about the elm and had no record of it on site. I did, however, manage to find 3 that were still alive, like this one on the right. But I think this shows that by accurately keeping records and maintaining relationships with people participating in campaigns, it will facilitate long-term monitoring to be able to see the performance in the long term. And then another way that the data could be used is to look at mature surviving elms. So from the data set, I was able to map the distribution of all elms that were either over 30 years old or described as mature. And that's the graph on the left. And then on the right, I filtered that further down into the mature British elms. Projects that have focused on propagating and planting out these elms have included those like the Great British Elm Experiment, again carried out by the Conservation Foundation, which propagated cuttings from a number of parent trees which were appearing to have survived the disease and so may or may not be resistant. So this is a great way to test it. And here I managed to plot maps for each parent tree And I know the dots are quite small, but there are a few that have been reported to be dead. And so over time, it will become clear whether these trees have resistant traits, and that would be a great example of native trees which are exhibiting resistance. So to summarise, I think my project demonstrated that accurately documenting elms and their locations can support long-term monitoring and future research on elms. This will also be dependent on collaborating and maintaining relationships with people participating in planting projects. From the data, you can also identify and track trees of interest, whether it's an ornamental trait, a climate tolerant trait, or resistance to diseases, not just Dutch elm disease, but other pathogens that affect elms. And then these could be used in future breeding work. I think it's also important to note that there were just 5 cultivars that were planted widely, and so it will be important to broaden the range of elms being used, but this is also dependent on availability and supply. So the records from this project are going to be compiled into an accessible database, and Richard, my supervisor, has also set up 2 iNaturalist projects, one for elms in the UK and one for planted elms. So I recommend that everyone checks these out and, if possible, submits their elm observations there as a great way to share data. So thank you for listening. A huge thank you to my supervisor, Richard Buggs, and Mohammed, who was a postdoc on this project, David Shreve and the Conservation Foundation, who are project partners, and everyone who provided data. I didn't have space to include everyone, but there are over 29 different sources, and I wouldn't have been able to do this project without you. So thank you.
Fantastic, thank you, Catherine. That's a huge amount of work in 6 months, and seems it's quite a challenging thing to sort of track down these projects that have happened in the past. Following on from that, because these projects are interlinked, we're going to move straight on to Richard Buggs, who is a research leader at Kew and is leading on this project for us at Kew Gardens. And Richard is going to be giving us an overview of the work on elm genomics. So I'll hand over to you, Richard.
Thank you, Louise. Good morning, everyone. Thanks for coming to this webinar. I'll be talking about some of the genomic analyses that we've been doing on some of these elms that have been talked about already. And as has been mentioned, this is in collaboration with Joan Webber. And Mohammed Vatanparast is the postdoc who's been working on this project. All of the analyses that I'm going to show you today were done by him. And we've also had the privilege of working with Juan Martin at the UPM Madrid, who supplied a lot of samples, including lots of hybrids that they have in the breeding program in Spain. And you'll see more about them shortly. So we took a whole genome sequencing approach, and we did this on 180 samples of elm, and we did them at 20x coverage. That meant that across the whole genome we could identify with some confidence the single nucleotide variants. So this could not have been done before, because the technological advances simply weren't there. It would have been prohibitively expensive to do this before now. And also we've only recently, as a scientific community, gained chromosome level reference assemblies for elm that we've been able to use. And most of what I'm going to show you today is a principal component analysis to summarize the variation in the samples. This is a method to try and boil down all of the variation across the whole genome as much as possible into 2-dimensional figures. So here is the first and second principal coordinates of our data. You can see this kind of triangle shape. And what we're finding is Ulmus minor is over here at one corner, Ulmus glabra at another corner, and Ulmus pumila in another corner. I'm not showing you Ulmus americana or Ulmus laevis in this picture because they are quite different to the others, and that if I included them, what you're seeing here, which is probably of most interest to us today, would all be kind of a bit scrunched up. You can also see various Asiatic species are clustered in the middle. Now, that's not because their variation is intermediate between these, and we can see that when we look at principal coordinates 3 and 4, those pull out the Asiatic species. So the Asiatic species are quite different. It's just that because there are not many of them in the main PC1 and PC2, they're not coming out as very different. Reassuringly, we find that known hybrids are coming out where we would expect them to come out. So, for example, Fiorente is known to be a hybrid between pumila and minor, and it's coming out in between the two. Similarly for Den Haag, Patriot, which has 4 different species within it, is coming out sort of where we would expect it to. Wingham, which has minor, pumila, glabra, and wallaceiana inside it, comes out kind of intermediate. Sapporo Autumn Gold and New Horizon, which are both known to be hybrids between Davidiana and pumila, are coming out between those species. So it all seems to make sense. Here's a family tree for a cross that you're going to hear more about in the next presentation by Cas Gudgeon. It was a cross done by David Hurling between Wingham and Ulmus minor at Tong Mill, and you can see the different parents of the cross, and some open pollinations also occurred in the origin of Wingham. Well, if we look on our PCA, we can see the parents here. We had a glabra component, a pumila component, a wallaceiana component, and then this is one of the parents of Wingham, FL493. 1038 was open pollinated to produce Wingham, and it's interesting that Wingham has moved slightly towards the middle, not simply towards Ulmus minor. So we suspect that the other parent of Wingham was also a complex hybrid. And this is where Tong Mill falls, which is the British elm that David Hurling crossed with 493. You'll hear a lot more about that from Cas in a moment. I want to focus now on this line that runs between Ulmus minor and Ulmus glabra. And I'm going to use the term Ulmus hollandica in a very broad sense for things that are hybrids between the two. Here's something called an admixture analysis of all of those trees that run into that line. So some of them are pure minor, they're just orange. Some of them are pure glabra, and then you can see some of them have admixture between the two. Some of them just with a very small component of glabra. And that maps directly onto this line that we can see in the PCA. So up here we have pure Ulmus minor. Some here have sort of less than 10% Ulmus minor towards this end. Then we have admixed individuals, and then we have pure glabra. And here we are just sort of zooming in a little bit on those individuals that we've included in this admixture analysis. So you can see a range of names in there, some of which many of you may recognize and that we've heard about before in this talk, these talks. I'll just look very closely at the cluster that includes the Ulmus minor end of this spectrum. At the very far end, we have a cluster that comprises some Ulmus procera from Matthew Ellis's collection, 4 Ulmus minor individuals from Galicia, and 2 Ulmus minor from Brighton. We think that this is probably the Atinia clone, the Ulmus procera in the strict sense, which is thought to have come from Galicia to the UK, brought to us by the Romans, of course. We then have separated from that a cluster of Ulmus minor in the broader sense. Then towards the edge, we have some individuals with a little bit of glabra, and then down here we have individuals that have much more glabra within them. And some of them are called Ulmus hollandica, which is what we would expect them to be called. Now, Selon Murrell named 63 different species of UK elm, and we had, not all of them, but we had some of them in our sample. 11 of them were supplied that had been verified by the experts, Brian Evesham and Alex Prendergast. And we can see that most of them are falling into this broad cluster of Ulmus minor, and 2 of them, as expected, fall into Ulmus glabra. Selon Murrell said that they were, closely related to Ulmus glabra. So what these species names seem to be telling us is they're useful labels for certain forms of Ulmus minor, but I think it's clear they shouldn't be viewed as species in the same way as Ulmus minor and Ulmus glabra and Ulmus laevis, etc., are viewed as species. And if we look very closely at the cluster where the Selon Murrell species are falling, we can also see that they're falling with other individuals from the UK and indeed from France that have been labelled as Ulmus minor. Also note that Reddii and Gudgeonii fall very close to this cluster as well. And here are the 2 Selon Murrell species that fall near glabra. They are slightly differentiated, but within the realm of what we would call glabra. Now, 7 Ulmus minor clones were released from Spain as selections of Ulmus minor from the wider environment that have survived Dutch elm disease. They were released a few years ago. The most famous of them is Adamus. In our results, Adamus seems to be 27% Ulmus glabra in its genome. It falls well within the range of individuals in the PCA that are integressed with genetic variation from Ulmus glabra. The same is true of Dehese de Amaniel. It's 18% glabra. And then Retiro and Dehese de la Villa are both, we think, pure minor. But it's these 2 that are the ones that are most resistant to Dutch elm disease. So throughout the talks, we've tended to call Adamus Ulmus minor. Actually, it might not be Ulmus minor. We're still checking this result. It was a surprising result, but at the moment, that's what our data seem to be saying. There's also some new light on other accessions. So Ulmus minor Amsterdam and Ulmus minor Beer Schwartz both seem to also have considerable amounts of glabra in them, and so perhaps should be seen as hollandica rather than minor. We had a collection from Frank Matthews that was called Dodwell, Ulmus procera Dodwell normal leaves, and one from Staffordshire. Those don't seem to be Atinia, they seem to be minor introgressed with glabra. We also looked at the range of individuals between Ulmus pumila and Ulmus minor, and again we found evidence of introgression of pumila into minor. And interestingly, some of these were things that have been identified before as minor or even as hollandica. So perhaps there is a component of pumila in them. We also looked at the genome-wide pattern of genetic variation in Ulmus minor and Ulmus glabra, and very noticeable that Ulmus minor contains considerably more variation. You'll see the scale for Ulmus minor goes from 0 to 2,580 SNPs per megabase, whereas in Ulmus glabra it's only up to 522. So we're finding a lot more variation within Ulmus minor than within Ulmus glabra, and that's in agreement with a previous study by Ewan Bowditch, I believe. So here's a hypothesis about what's going on with UK elms. I think we have a little cluster that is Atinia. Then just below a Spanish cluster, we have what is perhaps a native British cluster of Ulmus minor in the broad sense, including many of the Selon Murrell species. Then the edges of this have variation due partly to introgression from pumila and partly to introgression from glabra. And then we have a whole range of elms that contain more glabra. So in conclusion, admixture between minor and glabra may have been more widespread than we imagined. Ulmus minor seems to be more diverse than glabra in Britain at the genetic level. We've placed the Seller species firmly in minor, or some of them firmly in the minor cluster. We've shed new light on the origins of some widely planted clones, including Adamers. Some living collections contain mislabeled specimens. I haven't done a slide of that, but we did uncover many cases of that. And in conclusion, DNA variation within elms in Britain is clearly higher today than it's ever been in the past due to all of these introductions. So there's lots to be encouraged about for the future of elm in Britain. I'd like to thank everyone who supplied samples. It was a large number of people. And I'll just remind you, Mohammed Vatamparast is the postdoc who did all of the analyses that I have just shown you. Thank you very much.
That's great. Thank you, Richard. I think this really demonstrating how genomics work can add to what we know about these species and these clones and crosses. And we're going to stick with that that area now, and I'm going to hand over to Cas Gudgeon, who has been working with the team at Kew, also on genomics, but looking at genes involved in Dutch elm disease resistance and using the family from across pollination. And Cas is another of our interns who's been working on this project for the last 6 months, and this is just coming to the end of her project. So Cas, I'm going to hand over to you.
Good morning, everyone. My name is Cas, and I'm an intern at the CFP. So for the last 6 months, I've been working at Kew, and what I've been doing is some computational work on a family of elm trees, and I've been looking for genes that are involved in resistance to Dutch elm disease. So I'll start by introducing you to the experimental setup, and I'll give you some context about where the data comes from and the techniques that we use to look at the data. So around 10 years ago, David Herling crossbred an elm cultivar called FL493, or Ulmus wingham, which you can see on the right here, with a field elm. And David was really passionate about elm trees, and he really wanted to try and restore them to the landscape here in Britain. And although wingham is considered very resistant to Dutch elm disease, it doesn't really have the classic appearance of field elms that so many people love about elms. So he brought up this large collection of saplings with the hopes of creating a tree that combines the resistance of wingham with the appearance of field elm. And if you look at this family tree here, we have wingham at the bottom. And wingham is a really interesting cultivar because it's the result of a 4-way admixture between 4 species of elms. So it contains genetic material from 2 European species, which you can see at the top on the right here, Ulmus glabra and Ulmus minor. And it also contains genetic material from 2 Asiatic species, Ulmus comella and Ulmus willichiana. And so David tested these saplings for resistance to Dutch elm disease. So what he did was he inoculated them with a very aggressive strain of the fungus that causes Dutch elm disease. And after a period of about 12 weeks, he measured defoliation. And we considered about 25% defoliation to be the threshold at which an elm was considered susceptible to the disease. And the results were actually a very mixed bag. I think about 26 elms were considered resistant by that threshold, and about 34 were showing quite considerable signs of having succumbed to the disease. So the technique that we use to analyze the genetic data from these trees is called chromosome painting. And in our case, what that means is we look at all 4 elms that come from all 4 of those species that contribute to the genetic admixture, and we look at their chromosomes. And then we look at the chromosomes in the offspring. And looking at the chromosomes in the offspring, we try and figure out how to trace back every part of every chromosome to one of those 4 species, because we know that every part of the chromosome has to be inherited from one of 4 species. So we can paint all of the chromosomes according to that ancestry. And this is what that looks like. So these are the painted chromosomes for Wingham. And each of these bars represents 2 homologous chromosomes. So if you look at the bottom here, this is chromosome 1. And we have these blue regions have been traced back to Ulmus glabra, and these regions in orange have been traced back to Ulmus minor. This region here, for example, has been traced back to Pumila, and this chromosome here has all been inherited from Wilkiana. So we were actually able to do this for all the offspring and both of the parents and one grandparent in the Hurling cross. So it's just a very intuitive and interesting way of looking at how genetic material is passed on between generations. So for example, Wingham is the offspring of a clone called 1038, but since 1038 was open pollinated, we don't know who the male parent of Wingham is. But if we look at the painted chromosomes for these 2 elms, we can see some of the genetic material in Wingham hasn't come from 1038, so it must have come from the male parent. And if you look at chromosome 9, for example, in 1038, we can see that this whole chromosome has been inherited from minor. This blue chromosome has been inherited from glabra, but in Wingham, when we look at chromosome 9, we see this green region, which has been traced back to Pumila. So if we actually compare these sets of chromosomes, we can infer that the male parent of Wingham is a complex hybrid containing genetic material from all 4 of these species as well. And here are the painted chromosomes for some of the offspring. Here in this top right corner, we have the chromosomes of Ulmus minor at Tongue Mill. And since we have this classified as Ulmus minor, we'd actually expect all of these to be orange, but you can see some of the blue chunk, some of these blue chunks have been traced back to glabra. So that can mean one of 2 things. It could mean that either this tree genuinely has some genetic material which is traced back to glabra, and this is a hybridized elm, or because glabra and minor are quite closely related, it's difficult for the program we used to tell the difference between these 2 species in certain regions, where it doesn't really have enough information to distinguish between them. So some of these blue regions, I know it's hard to see, but they're very small and they're very wobbly. Not all of them, but that does tell you that there's quite a bit of uncertainty in those predictions. And then also, if we look at the offspring, you can see that the Asiatic ancestry has been diluted since crossing Wingham with a European elm. We can see that there's a lot more sort of blue and orange in the offspring and a bit less pink and green, which tells us the Asiatic ancestry has been diluted in these elms. So why do this analysis? What's the practical use of doing this? If we suspect that Asiatic elms are carrying resistance genes, then we can use our chromosome paintings to try and look for those genes. And we can compare the resistant elms and the susceptible elms and look for regions where we see that the resistant elms have inherited some material from the Asiatic species that hasn't been inherited in the susceptible elms. So in this slide, we can see on every chromosome where material has been inherited from Ulmus pumila in the resistant trees and the susceptible trees. So if we look at chromosome 9, for example, we can see that the susceptible trees have actually been the ones to inherit more material from Ulmus pumila than the resistant trees. And if we look at this next slide, this is looking at which chromosomes have this is if we look at chromosome 7, if we look at this region here, we can see that the resistant trees have been a lot more likely to inherit genetic material from Ulmus willichiana than the susceptible trees. And that tells us that we could potentially find a gene or a few genes in this region which are conferring resistance to Dutch elm disease. But what's important to bear in mind here is that this is a region that actually represents about 35 million base pairs. So although we've been able to narrow down our search from 1.8 billion base pairs to 35 million base pairs, it still leaves us a lot of work to do in terms of trying to find very specific genes. But it's a really good start for future research. And something else that we looked at as well was how do disease symptoms correlate with overall ancestry proportions for each species? And by global ancestry proportions, we just mean how much genetic material has been inherited from each species overall. So for the Hurling cross, having one parent which is almost minor means that the global ancestry proportions are at least 50%, and in this case they're actually closer to around 70% on average because of some of the almost minor content in Wingham. But interestingly, we didn't really find a strong relationship between disease symptoms and global ancestry proportions. Actually, if you look at these plots, ancestry from Pumila, for example, is slightly correlated with more defoliation, and ancestry from minor is slightly correlated with less defoliation, which is sort of the inverse of what you would expect, because we know that Pumila is resistant to the disease and minor is susceptible. So from this, we can it kind of suggests to us that disease resistance seems to be conferred by a very small number of genes, rather than being something that is influenced by a really large number of genetic factors. So that's just a really quick overview of the work that I've been doing. Thank you so much for listening. And I'm very grateful to David and Fergus for providing Kew with a really rare and invaluable resource for elm research.
Thank you so much, Cas. It was really interesting to see all of those chromosome paintings, which really shows what a mix there is across these hybrids. So we're going to move on to our last talk now, and now for something completely different, because this is a social science project. It's another of our interns, Jack, who has been working with Forest Research and with the University of St Andrews. And Jack has been working with one of our PhD students, Fritha West. We'll be having a webinar with Fritha and a couple of the other PhD students from the Centre for Forest Protection later in the year. So if you've joined our mailing list, we'll be able to send you details of that in the autumn. So I'm going to hand over to Jack now, who's going to tell you all about land manager perceptions of elm loss and restoration.
Lovely stuff. Thank you, Louise, and thank you everybody for coming. So as Louise said, I'm Jack, and I'm an intern based at Forest Research and the University of St Andrews. And today I'm going to be presenting on perceptions of elm loss and restoration. So this is part of a wider interdisciplinary project looking at the social acceptability of lost tree species restoration. So this is supported by an amazing team across Forest Research, the University of Andrews and Kew Gardens. Of course, Fritha, who is in the same room as me today, so thank you for that. So this project uses 3 case studies chosen for their different relationships with elm. The Wye Valley, the centre of my case study, was chosen in part because of its large-scale historic loss of elm and other tree species, as well as high forest cover, large forestry sector, and a long-standing cultural connection to trees, as well as numerous ongoing restoration projects and some locally significant tree species that own, with the apple orchards and the native black poplar shown on the slide there as really good examples. So this project set out to look at land manager perspectives, but from early engagement, this expanded to encompass a wider range of stakeholders as we saw that there's more people involved. The objectives of this project were to explore perceptions of tree species loss and its impacts, and to identify motivations, challenges, and opportunities for restoration. So this was achieved by conducting 14 in-depth semi-structured interviews, both online and during fieldwork in the Wye Valley this January, with interviews subject to preliminary analysis, which I'm going to be presenting on today to give you a bit of a snapshot of the project. So who did we speak with? So as part of engaging with wider stakeholders beyond traditional conceptions of land management, we spoke with some amazing individuals, from those involved in decision-making, those who actively manage trees and engage in research, and those involved in harvesting and the use of wood, such as sawmillers and woodworkers. So we had just about over 50% of our participants from a productive land use background there. So what do we mean by loss? So it's important here to note that perspectives of loss, it's not the same as local extinction, but rather what individuals consider loss means to them. This tends to cut across biological, functional, cultural, and social forms as the basis of our project. So elm, obviously, is our main case study, species that we set out to speak with participants about, with them describing different relationships between wych elm and English elm here. So wych elm, more small scale relationship, and it's less visibly present in the landscape and confined to woodlands. And English elm and field elm, more recent loss experienced through Dutch elm disease and its impact on the farming landscape. Beyond elm, participants identified a range of species with native black poplar and the heritage orchard varieties, really key areas of local concern, as well as the ongoing loss of ash, larch, and the potential future loss of oak as well, a real concern. So quite a high level of awareness around tree species loss in our participants. So how did our participants describe loss? So they described themes of change. I think that change is really key. So they described changing landscapes. So obviously you've got that loss of hedgerow trees and that classic elm landscape, which people really drove home. One of our participants quoted Orwell, who described it as a magical elm landscape, a magical elm forest. And this is being replaced with dead trees, standing sentinel, and now a complete absence from the landscape. I think that's a really quite otherworldly photo there. We've got changing landscape, changing relationships, sorry. This is past, present, and future. And I think an elm chair is a really lovely example of shifting use, something that used to be fairly common and is now sort of a rarity with sort of hopes for revival for the future. And in terms of changing memory, we've got intergeneral changes in memory and ownership and recognition. So how people see trees is very different. And if you haven't seen it, it's hard to conjure up what you're lost, really. So in terms of the impacts of loss, so stakeholders identified economic, ecological, and sociocultural impacts derived from these changes. It's important that we note that these aren't siloed and they do cut across different groups, but for ease of communication, we've sort of categorized them a little bit. So in terms of economic, you have both the cost of managing and then the resource itself as well. So for example, the cost of managing loss, with one participant stating that local councils were having to fell healthy ash, as well as, because it's just more cost effective, as well as sawmills saying that importing elm from Scotland and further abroad is just not, it's just unsustainable and expensive. In terms of ecological loss, species reliant on elms, such as the white letter hairstreak there, as well as hundreds of other dependent species, has had a massive impact on their habitat. And in terms of sociocultural impacts, we've got a decline in presence and everyday use. I think the tools there and also kids around the tree, is just that fading recognition, leading to shifting baseline and eventually cultural amnesia. So in terms of motivations and restoration outcomes, these aligned broadly around functionality, biological diversity, aesthetics, and cultural value. Also important to note that these aren't exclusive and they do cut across each other as well. So in terms of functionality, we've got uses for elm. So we've got the beautiful waning edge cladding there, as well as timber framing. So this like practical purpose and practical use is a real driver for restoration and what people want to see as successful restoration. In terms of biological diversity, so supporting those native species, the white letter hairstreak comes back again, as well as all of the other dependent species. So really providing that habitat and connectivity across the landscape as is associated with the elm hedgerows. In terms of aesthetics, you've got that classic figure of 8 shape and the classic elm landscape shown on an image a couple of slides ago. And in terms of cultural history, we've got that sociocultural connection to elms. So in different uses, you've got the pilot gig boats, you've got from Cornwall, you've got green woodworking, so and its everyday use in bowls, tools, and it's just our presence in our day-to-day lives. And that's what people really want to see. I think people want to experience it. And it's important that we note here that restoration, it's not static, it's not a return to the past, but it's very much forward-looking and evolving, and people want to sort of see that in the future. So we found that challenges break down into organizational challenges. So that's around sustaining long-term effort. And we found that quite often the restoration efforts depend on like one or two linchpin individuals. So it's really about supporting them and creating broader networks through knowledge sharing and community development. In terms of financial challenges, we need to meet the size of the restoration challenge with the right financial resources, joining up projects and extending them to create more long-term forward-thinking restoration networks. In terms of public attitudes, so this, I think a lot comes down to building trust between land managers, policymakers, and researchers, so the guidance is seen to address the wider needs of these communities and what they're asking, as well as sort of making it clear that there's no invested interest. That was a real concern, as well as addressing different cultural differences currently preventing restoration between different groups. So really building community is really key there. In terms of management challenges, these involve managing our woodlands more actively to reduce browsing pressure from deer and squirrels, with one participant stating that, in fact, deer is the largest threat to elm and ash's continued presence in our woodlands, not disease. So that's a really key issue. And it's also important that we utilise the technology where available. So in terms of opportunity for the restoration of lost trees, there's plenty of opportunity with stakeholders being interested and really enthusiastic about elm restoration, and pretty much willing to put the effort in, even if they're not being logistically supported currently, showing that, in fact, it's not a lack of interest or enthusiasm, but it's generally a lack of means that's stifling current restoration efforts. Alongside this, there's a high level of awareness around the need to future-proof woodlands and the UK's timber industry, especially concerning climate change resilience. And I think it's really cool that elm was seen as being potentially useful for this, especially in terms of agroforestry, promoting woodland diversity, as well as acting as a framework for other species in the for restoring other species in the future. So overall, this project has given us a snapshot of stakeholder perceptions of tree species loss and restoration, with this cutting across species, time and space. There's an interest and a willingness to invest in restoring elm and other locally significant tree species, such as the native black poplar. And despite there being logistical challenges at current, there is the innovative spirit and the willpower to achieve this restoration. And I think this is a really lovely quote that we can end on, that, there's a lot of future for elms if we can, if we can make them viable again and if we meet the needs of the restoration challenge. So yeah, I'd like to say thank you for listening, and I'd like to thank everybody that we've engaged with during fieldwork. I'd like to thank the referrer as well for her help during this project. Thank you all for listening. Cheers.
Brilliant. Thank you so much, Jack. And that helps really put, some of the other work that's going on in perspective and how it impacts on people and communities. So I think that's a really great thing to add to our talks today. Right, I'm going to start with a couple of questions we've got coming up, Joan, about the beetles. So one question is, how far do beetles tend to disperse from breeding trees, and what are the factors that affect that dispersal?
Right. Well, this question of how far any insect disperses is always a bit of a vexed one, I think. But the sorts of things, so people have tried experiments, releasing beetles from a spot in the middle of nowhere and then trying to recapture them, which is always difficult because they get diluted. But I think it's safe to say they probably fly a few kilometres, maybe 2 or 3, and of course they may be wind assisted and taken longer distances as well. So probably 2 or 3 kilometres are at the limits of it, and I would definitely suspect that from the way you seem to have this difficulty in beetles finding trees that are dying of Dutch elm disease, and particularly early on in a little local epidemic, they don't get noticed, they don't get found, not enough beetles find them to breed in them. So I think it's relatively limited, and it's mainly helped by people moving around firewood, I suspect. So the factors that affect them, temperature. Temperature is really important. Supposedly it's something like has to be about 16 degrees external temperatures before they start to emerge. And really it needs to be probably over about 21 degrees before they start flying. And so you can see certain years and certain springs where you get a series of hot, warm days, and that seems to synchronize the beetle emergence and dispersal. And then almost a month later, you'll start to see the symptoms appearing of flagging sort of twigs and small branches in the crowns of trees. So it's about temperature, and of course they fly towards the light as well.
Brilliant, thank you, Joan. And another question, this was from Seb Crichton, who said, are there any differences in how the two beetle species feed and breed? So would one be more of an efficient vector than the other?
The larger elm bark beetle seems to be the more efficient vector. And by that I mean a more effective vector. It's more likely to carry spores of the fungus. But I don't think that's just because it's bigger. I think it's really where they breed in the bark. So the smaller elm bark beetle, Scolytus multistriatus, tends to be, when the two are there, the smaller bark beetle tends to be pushed out and breed in the smaller diameter material. So in the branches, in the twigs, and these dry out more quickly, so the fungus is less likely to survive. Whereas the larger elm bark beetle, Scolytus scolytus, tends to breed in the thicker bark, and that's where the fungus grows and sporulates. And at the end of the day, the thing that's most important, even though the fungus really is throughout their galleries, again, it all happens really at the end point. The larvae make a little chamber, it's called a pupal chamber, and they pupate in there and they shed their skin so they don't carry over any spores. So what is really important is how well the fungus then colonizes the surfaces of that pupal chamber for the new beetle to pick up the fungus. And it's most likely that the fungus grows the best when it's in the phloem tissue, not when it's in the outer bark or not when it's in the sapwood. So where that pupal chamber is really dictates how well the fungus grows and how likely that new generation of beetle is going to be reunited with the fungus, so it carries it out. So the larger elm bark beetle tends to breed in thicker bark. It's sort of juicier stuff for the fungus, and that's why it's more often carries out the fungus as well. I hope that's clear.
Yeah, so it sounds as if there's actually a lot of interaction between the fungus and the different tissues of the tree and where the beetle is in the tree. Yeah,
Absolutely.
thank you for that. So question for Richard from Pascal, who said fascinating genomic results, particularly the marginal zone between Ulmus minor and Ulmus glabra. But just to sort of clarify, where would you draw the line between introgressed individuals and hybrids?
Well, thanks. Yeah, I'm glad you enjoyed the genomic results. I think it's hard to draw a line, and it becomes quite semantic, really. So some people would use hybrid to refer to anything that's got any introgression within it. Some would want to just use it for a very early generation, even just the F1, and then call anything else a backcross. So I think it's hard to draw a line. And I think different people with different priorities will sort of draw different lines from the results. So if there's somebody who only wants to be growing 100% pure Ulmus minor, then they might take a different view to someone who simply wants to grow something that approximates to Ulmus minor in the landscape and will be more resistant to Dutch elm disease.
Thanks, Richard. And a question related to that from Max, who said he does think that hybrids and introgression are common in England, but do you think the view of it could be exaggerated by assumptions about the origins of the DNA? So I'm assuming the sort of parental origins of the DNA is what he means by that.
Well, it's not the case. So sometimes in the past, people would use a set of markers developed for one species and then use them on another species and the species they were developed on. So we're not doing that here. We've got whole genome read data. It was mapped to the Ulmus americana genome assembly. So that's pretty unrelated to both glabra and minor. So I don't think there's anything in our method that is going to be exaggerating the level of introgression that we're finding.
Thank you for that. Jack, a question for you. It was interesting that you were saying that there was enthusiasm in communities even where there wasn't support. What sort of things do you think would help communities to be looking after their elms and doing the sorts of things that you were seeing people doing? What sort of support do they need?
Yeah, absolutely. I think a key part of it is honestly knowledge sharing and networking. I think that's something that across my case study and also just like wider case studies that we've been involved in, is that people really want to network and be able to share their experiences working with elm. Yeah, so I think knowledge sharing is key to that, as well as sort of connecting people with sources of funding. I think it's all about that interconnectivity, and I think that's something that comes time and time again, really.
Brilliant, thank you, Jack. Cas, I've got a question for you, which is a little bit about the experience of the internships, because obviously it looks from your charts and things as if you've been using a complex program. And I was wondering if you knew anything about using that type of tool before you started the internship, and if not, how you found the experience of learning it.
Yeah, I came into the internship with some experience of using computational clusters and using the Bash programming language to navigate Linux, but it was definitely a very steep learning curve, and I pretty much just sort of had to teach myself things and sort of figure things out on my own, because I think especially with this kind of thing, you very quickly get to a point where people can't really help you because what you're doing is so specific to one program and one problem, and it would take them ages to try and catch them up and get their insight. So yeah, I was just sort of I find that what was really helpful is computational biology is very open, and I was actually able to reach out to multiple developers who had created the software I was using. And in some cases they had developed the software over 10 years ago, but if I emailed them, they would respond to me within a few days saying that they were very interested in what I was doing, and they would give me very clear instructions on how to use the tool that they had made. And so I think what was really helpful was just software developers who were just really interested in the research I was doing. And I think that something I've noticed is that scientists are very collaborative and very open to correspondence from even if they've never heard of you and they have no idea who you are. So that was great. Yeah, but it was just a lot of tinkering and being patient and trusting that it would work out eventually.
Thanks, Cas. So it sounds as if in the process of it, you become an expert in what you're doing, more than perhaps anybody else in your team, which is amazing.
Yeah, you definitely.
Yeah, thank you. And Catherine, I was really interested in what you were saying about the project with schools, and then trying to trace what has happened to those elms afterwards. And obviously there are lots of organisations and charities that work with schools to encourage things like tree planting. And I wondered if you had any advice for anyone who's setting up a programme like that, to make sure that it's also useful for researchers in the future based on your experience.
Yeah, so I would say that one of the first things that would probably be helpful is once you've supplied a tree, once you've sent an elm to a school, to get information back about where they have planted it, and when, as not just so that you know where it is, but as like a first kind of way to initiate their role in looking after the tree, because that would also encourage them to maybe keep their own records so that they could be kept for longevity, and also to make sure that if kind of management changes hands, so for example when schools would change site, or where like the person who had organised to plant the elm left the school, to kind of pass on that knowledge to the people taking over, because obviously it takes a long time for a tree to reach maturity, so it is likely that there's going to be a changeover in the people involved during that time. So trying to keep the kind of flow of communication going regarding that tree would be important.
Thank you. Yeah, so perhaps handing over responsibility for the project at both the initiating organisation and at the schools, to make sure that there was ongoing communication about it. Thanks, Catherine. We've got an interesting question here from someone who says that on the Isles of Scilly, we don't have Dutch elm disease, it says yet in brackets. As part of our landscape recovery project, we're working to improve biosecurity. So this is probably mostly a question for you, Joan, as someone who knows a lot about this area. And they're asking, what would be your top tips in terms of biosecurity to prevent the entrance of Dutch elm disease?
Yes, thank you. Well, funnily enough, I was having a conversation with Kelvin Hughes, who works for APHA last week, who was telling me about this. And I would say the absolute top tip is be very careful about any movement of firewood being brought onto the islands from elsewhere, because I think that's really where you see programs break down, or protection programs around controlling Dutch elm disease. In England, for example, or Scotland, it usually can be traced to somebody having a little consignment of firewood sitting in their shed or in their back garden or something like that. So that, I think, is really the key, and education and that sort of thing. But I think that's one thing that really has to be policed the most closely of all. Because but the other thing that's interesting, and again, I did have a chat with Kelvin about this, it would be interesting to know whether there are any of the native bark beetles on the island, because the beetles can exist without the fungus and make use of bits of windblown elm or whatever to breed in. But I think as far as bringing the disease in from elsewhere in Britain, it's all about the firewood. To me, that would be the thing you really need to keep a close eye on.
Brilliant. Thank you, Jen. That's really useful advice, I think. We have another question here on the chromosome. Oh no, sorry, that's just it's a suggestion. There's also some suggestions on Slido, so just to make you and then a follow-up question here, actually, Catherine, on where it would be useful to keep the records on school projects.
Well, so as part of my project, we're planning to publish the data in an accessible database. So hopefully, I don't know exactly how it would work, but hopefully more information could be added to that over time. But like I mentioned with like the iNaturalist projects, you could take a picture of the tree and put it on there. But I also think it would be important for the individual organisations and schools to have records of it themselves. And I know that might be hard to implement, and it'd be I'm not sure exactly what the best way to do it would be, but maybe you could incorporate it into the curriculum, like every year you do an experiment where you take measurements of the tree, and then that would go into some kind of record system.
Thank you. I've got a question, Joan, from Jonathan Norris from Forestry England, who manages a woodland with lots of different, he said, P1978 elms. Some are dying from Dutch elm disease and some are healthy. If the beetle population is now high, is it likely that they'll all die over time? We're bearing in mind that there are some that are clearly quite healthy now.
That's a really interesting question, and simply I don't know the answer to it. I think some of the surviving elms, some of them that we see in the countryside, are resistant, not just have this field resistance, so resistance to the beetles, but probably have resistance to the fungus, Ophiospermum novoalmi, as well. And of course, some elms may well have a combination of both, a bit of resistance to the fungus and this field resistance to the beetles, which sort of work together, I'm afraid. So I would say one of the sites that I talked about where we looked in detail at the bark beetles and the sort of trajectory of the disease over time. One of my sites I go back to, and virtually every elm, I think, yes, 95% of them are dead, the big ones. It's only very small ones remain, apart from one group of elm trees in the hedgerow. And they are just looking as good as ever. And I keep thinking to myself, I am sure they must be resistant. But the way forward would be to try and propagate and challenge them with the fungus and see if they are. And maybe also do some genomic work as well. So I think some of them will be resistant and some of them won't be. So it's unraveling that. So it's great to hear you have elms in the woodland, and I really hope that some of them will go on and survive and prove to be resilient.
And a follow-up question to that would be if land managers removed all of the bark or the trees with breeding beetles in them before emergence, can you stop a population spike or decrease the impact?
Yes, yeah. I mean, that's really the main plank of Dutch elm disease control, taking out the trees either before the beetles get to breed in them or before any beetles emerge from them. And so it's, yes, you would have to do that, and that could be quite hard work. So it's all around removing that bark. But then if the trees do die and they do hang around and they don't get used by breeding material, they don't stay there available for the beetles. The window between the elms being suitable for breeding by the beetles and not being breeding is relatively short. It's probably a couple of years, because although we talk about beetles breeding in dead elms, the bark has still got to be alive for them to breed in that bark successfully. Once that bark has died, then it's useless to the larvae. It's not nutritious anymore, gets invaded by other things. So the ideal would be to either remove the bark, or maybe even gather all your material together and try and cover it with tarpaulins or something like that. Anything that you can do to sort of delay or hinder that whole process, I think.
Brilliant. Thank you, Joan. Question, probably this is more for you, Richard, I think. Is there a route to a Dutch elm disease resistant elm that looks like the English elm Atinia via traditional breeding, and what would we need to do next? So I know it's not strictly genomics, but it's thinking, yeah.
Yeah, but I think our genomics results do shed a bit of light on that. So I think one thing is it's very interesting that a lot of the selections or cultivars that were thought to be pure ulmus minor and resistant turn out to have some level of introgression in them from another species, which to my mind, is suggesting it's very unlikely that we are going to find something that's pure Ulmus minor that is genuinely resistant. So it will need genetic material from other species. Will we be able to do that with traditional breeding? Well, that's what David Hurling was setting out to do by crossing Wingham with Tong Mill. What we need to do next is do another generation from his. So what we need to do is take the best trees from his cross and then cross them and get an F2 generation, because then it's possible that some individuals in the F2 will have 2 copies of any genes associated with resistance that might be beneficial, and also there'll be another round of meiosis, so that the chunks in the chromosomes will get that much smaller, so it'll be easier to map where there's resistance. And so there might be a route through by essentially continuing what David Hurling was doing. The other approach might be to try to use genetic modification. So if we could identify, if it were the case, and we don't yet know if it is, but if it were the case that there are just a few genes that can confer resistance, we might be able to use gene editing or GM in order to introduce them into the Atinia clone. And of course, if you want something in the Atinia clone, because it's not sexually reproducing, it would have to be some sort of GM. It's not amenable, as far as I'm aware. Correct me if I'm wrong, anyone. It's not amenable to traditional breeding because it's clonally propagating.
Thank you, Richard. And a follow-on to that, so obviously you mentioned gene editing there, but are there opportunities for sort of combining traditional breeding with the genomics information, so having genomics informed breeding for resistant elm?
Yeah, so if we do manage to identify specific locations in the genome associated with resistance, then if we are breeding, we could select early for them and just grow trees that already have that, because obviously with a traditional breeding approach for Dutch elm disease, you've got to wait quite a long time before you can do an assay to see if they're really resistant. But if we knew that you needed to have a certain gene, we could take loads of seedlings and just pick out the ones with that chunk of genome for growing on, which would reduce the amount of space and the amount of effort needed to do the selections.
Brilliant. Thank you very much. So I think we might have time. I've got a note here from Elizabeth saying she's got a question for Joan, but it's really complicated. Elizabeth, I don't know if you want to contact Joan offline with that one, or you could turn your you could possibly turn your camera and mic on now, because I've enabled that if you want to.
Yeah, hi. Yeah, it just wouldn't let me write it all in the chat because it became long. But it was really about Laevis and the fact that we're seeing infection in Laevis. So it was really, is it because the beetles are now moving over to feeding on Laevis because there's not enough other elm for them to feed on, and therefore Laevis is getting a higher inoculum load of the pathogen? Or do you think there are changes in the pathogen itself that means that Laevis is more susceptible to a sort of new strain of the pathogen?
I don't really think it's due to a change in the pathogen. When you inoculate Laevis, it is very susceptible, full stop. So it will be so being more susceptible wouldn't make much difference, in my view. I think it's a bit of a numbers game with Laevis. As long as you're a certain distance away from the beetle, so beetle numbers are diluted before they get to you or find you. I think that and this natural deterrence, I think, all works in your favour. I just think once you get these local build-ups, and so beetles are just flying short distances, then however much you get a proportion of the beetles avoiding and flying on, you still get quite a number of the feeding positions filled up. So I just think it's the law of probabilities. When we worked in the field before, this is years ago now, we found, working with Ulmus minor, we found if you looked at the number of feeding grooves there were and the numbers that resulted in infection, it was quite small. It was only about 5%. So it's not an absolute given. Just because a beetle feeds, it's going to transmit the fungus to the tree, because it depends on the number of spores the beetles are carrying and how long they feed for and how deep the feeding groove. So I just think what happens is there are enough beetles that will feed on Ulmus laevis, even though maybe the majority of the population don't like to feed on it. Some of them will be less discerning. Some of them will be happy to stick with what isn't the preferred diet, if you like. So I think it becomes a bit of an inevitability when beetle numbers build up and you have those sources nearby. Sure. And the other thing that quite interested me was I was expecting a bit like wych elm. Sometimes the beetles don't breed so successfully in wych elm bark, and also they don't like to feed on wych elm so much. But with some of the wych elm that we were seeing in Hampshire as part of these plantings, there were quite sizable trees with the stem diameters, and there was lots and lots of beetle breeding going on in there. So there seemed to be plenty of beetles available to feed, and then as the trees declined, they all piled in and started breeding in there. So once you got one Ulmus laevis fail, if you had others sort of close by, the whole thing started to implode a bit, if you like. Yeah. Glad I didn't have to type that into the chat either, Elizabeth.
Thank you, Elizabeth and Joan. I'm going to go to Max next, who had a popular question about wild survivors. I'm assuming that's the one you're going to ask, Max.
Well, actually, it was more a kind of reaction, and I was just going to confirm for Richard that he's quite right, that you can't really do traditional breeding with English elm, the Atinia clone, because it does appear to be sterile. I've never seen viable seed on it, and many respected ecologists have commented the same way. I think Oliver Rackham said he'd never seen a seedling of English elm before. So yeah, I think breeding is a kind of a bit of a non-starter there. And I can't help feeling that, in a way, David Hurling's ambition to recreate that beautiful figure of 8 shape is possibly a bit of an impossible task. And he wasn't breeding from, because he couldn't breed from English elm. And even with GM, I'm not sure it's a wise approach to kind of creating future resilience in the elm population. We'd be still relying on a clone to replicate a form that we have this association with, which I understand because it was such an important feature of a certain landscape. But it may be that is part of history now. And then just in relation to Joan, I don't know if you were implying that wych elm behave rather like white elm, but certainly my experience is that wych elm, curiously actually, is highly vulnerable, and that the number of survivors, and you described a nice example of finding survivors, I don't know if it was wych elm, but finding these survivors that you find up and down the country that we don't understand why. But for wych elm, a population that gets exposed to disease will suffer very, very badly, and the number of survivors is exceptionally low. I've got no evidence for this, but I will quote sort of less than 1%. So it's curious that a native species that you imagine might have some good genetic diversity and might stand up better to the challenge of disease seems to suffer very, very badly.
Joan, welcome to you.
Well, I suppose it's as far as Ulmus glabra is concerned, wych elm is concerned in the beetles, they're both natives, and I'm sure they've got their own relationship sorted out. What we've done is fling this non-native into the equation in the form of this fungus, Ophiostoma novoalmi, and that's what's really upset the apple cart, I suppose, up to a point. I was just going to go back to, I put my hand up because I was going to say, you can get pollen, you can, the pollen from English elm, and that can be used to fertilise other elms. And I know in the past we've collected that and supplied that to elm breeding projects in North America. So you can use English elm as the male, even though you might not get fertile seed. Something that I know Elizabeth Orton and James Brown are trying to explore this issue about: is the seed invariably infertile with Ulmus minor, or is that due to the fact that you just have collections of clones all growing, one clone growing close together, so you're most likely to encounter the pollen of yourself, and that may not fertilize you. So yeah, but you can use the pollen.
It seems to be English elm that's the peculiar one, because other field elms do seem to be pretty fertile. So you can do breeding with other clones of field elms. It's just the English elm that's the challenging one.
Thank you, Max. Right, we are at the end of the session now, but actually there's still a couple of interesting questions that I would quite like to come on to. There's one, so if any of our speakers do need to drop off, please do, and obviously any of our attendees, but it would be interesting to just cover these couple of questions. And one of them is, do we know what the genetic variation of the fungus is? And I know this doesn't relate directly to the work done on these projects, but I was just wondering if you knew much about that.
There has been a lot of work done on the genetic variation in the fungus and how it's changed as it's moved as a pandemic across the northern hemisphere. So it's an outcrossing fungus, and it goes through sexual recombination very frequently. So we know out there in the population it exists as a huge number of genotypes, and not only that, there are different subspecies of it. One that's come from North America and another that's come from sort of further to the east, probably Asia. And there are parts of Europe now where these two subspecies of Ophiostoma novoelmi are interacting together and hybridizing. So so you've not only got these two subspecies with a great deal of generation within them, but you've got this hybrid swarm now being created between these two subspecies as well. So it is pretty variable, and to some extent it's probably still evolving. And we've seen it's maintained its virulence really over certainly looking at populations in Britain since the start of the epidemic here. It's still pretty much as virulent as it was right at the beginning.
Thanks, Joan. And we've got a question about if we're introducing these different elm varieties, do we know much about the biodiversity value of the different elm varieties? Is that something that there's been work done on, or that would need to be taken into account if we're planting them?
I don't know whether I feel my voice has been heard quite a lot, but one thing, talking to different elm groups, and I thought they would favour more some of the elms that come from Europe and are pretty much maybe not pure Ulmus minor, but principally Ulmus minor, whereas some that have been derived through these breeding programs and made use of backcrossing or crosses with Asiatic elms. But I think people who are planting an elm have different priorities, and sometimes they want, and they're not necessarily bothered about its genetic background, it's how it looks. And I think for other people that priority will be more around what role it can play in biodiversity, and for others it'll be about timber. So there won't be one size fits all. And I suppose that our job is to try and get out more information about what some of these different cultivars can bring to people, and why they want to plant an elm, and whether this one is the best fit, regardless of whether it is an Ulmus minor or whether it's quite a complex hybrid. But other people may have a view on this, a different view, I'm sure.
Thanks, Joan. And one final question for Richard. With the work on sort of finding out the different mix of genomes, has it changed your perspective on what we consider to be native or not native in Britain amongst our different elms?
It's a good question. I think the amount of diversity that we're finding in Ulmus minor is suggestive of a native species. And if there really is if our signal of very small amounts of introgression of pumila in some of their genomes is true, then it suggests that pumila may have been around for quite a while as well, or at least trees were brought in from the continent that had a pumila component in their genomes. The whole native, non-native debate is one generally I try to avoid. Well, one thing I would add to the discussion is that it's fascinating that some of the most resistant relictual elms do seem to be hybrids between minor and glabra. But as Max has been saying, glabra itself is highly susceptible. So it's not that there's a gene in glabra that is making some introgrescence with Ulmus minor resistant. So that suggests that there's some sort of heterosis going on, and that somehow it might be beneficial to have two copies at a certain location in the genome, one from minor and one from glabra, or that there's some genetic material in glabra that is not present in minor, and in minor that's not present in glabra, and to combine that together is beneficial to the tree. I think that's something we probably need to explore, because otherwise it's a puzzle why two susceptible species, when combined, seem to be able to sometimes produce a tree that is a bit more resistant.
Brilliant, fascinating. Thank you, Richard. Right, well, I'm going to close the webinar there. Thank you so much to all of our speakers, and a really interesting set of talks there. And thank you so much to all of our attendees for taking an interest in the work that we're doing at the Centre for Forest Protection on elm. Apologies if I didn't get around to asking your question. So thanks once again for attending.